Today we'll be talking about molecular basis of inheritance. Now, why molecular basis of inheritance? So until now we've been studying Mendelian genetics and then the other types of genetical studies that came from Mendelian genetics, or based on Mendelian genetics we have different kinds of genetical inheritance patterns that we have studied. So other than classical Mendelian genetics, you have things like codominance or intermediate dominance or polygenes and other kinds of inheritance patterns that we have seen. Now the problem with that kind of inheritance pattern, or studying that kind of inheritance pattern, was that all of it was done not at the gene level. We did not study anything at the molecular level, nothing was studied at the cellular level. So the study was not done at a cytological level, but rather than at a organism level. So Mendel, for example, was working with garden pea. So he was working with the plant completely and not working with specific cells inside of the plant. So he was working at the organism level, and pretty much all of genetics works at an organism level. And although we have learned a lot from those studies, meaning that we know that there are factors, there are traits which get expressed, there are alleles, all of these things we have learned from that, but we don't have a molecular basis of it. So we don't have a cytological reference to it, and that is the reason why we study molecular basis of inheritance. And like I said, we are going to be studying the genetic basis of it, so the genes. Now a gene is the shortest fragment of DNA that can hold one specific information. So it is going to hold one specific information, and by its expression it is going to give rise to one phenotype. Alright, so it is the shortest length of DNA that is going to hold one specific information, and its expression is going to give rise to a phenotype. Now the term gene was coined by Wilhelm Johansen in the year 1909. Okay, now there are certain properties that a fragment of DNA needs to have for it to be called a gene. So a gene, for it to be called a gene, needs to have certain properties, and the properties are here. Now the first one is stability. Stability meaning that the gene needs to be stable, as in it needs to be passed on from one generation to another generation in the same form, or mostly in the same form. It can undergo some changes, but majorly it needs to have the same form and needs to be passed on from one generation to another generation, and that's why it's the stability. The next one is alleles. About alleles we already know that a gene is going to have a minimum of two or more alleles, and each allele is going to give rise to one specific phenotype. So if you are going to think of Mendelian genetics and think of the trait for tallness. Now tallness in garden pea was either dominant, which is the dominant allele which would give rise to a tall plant, or a recessive allele which would give rise to a dwarf plant. So it's the gene for height, and then it has two alleles, one dominant which is going to give rise to tallness as a phenotype, and then another recessive which is going to give rise to the phenotype as dwarfness. So each of the alleles give rise to a different phenotype. So it's not just that the gene needs to have alleles, it needs to have that each and every allele is going to give rise to a different phenotype. Now the third one is locus. This is again something that we have studied in Mendelian genetics, that the gene needs to have a specific location on the chromosome. So it needs to be present at a specific location on the chromosome, and that location is going to be the same in all of the organisms, meaning that if you take one population of a specific organism, if you take that the location of that specific gene in that entire population, you will find that it is there in the same location. Okay, so the locus is going to be stable throughout a population. The fourth one is the ability for it to mutate. Now that is what I was saying when I was talking about stability, that not just that it needs to pass on from one generation to another generation, it needs to pass on in the same form. Most of the times it can undergo changes, and that undergoing change can happen through mutation. So a gene needs to have the ability to mutate. Unless and until it has the ability to mutate, it cannot be called a gene. Now the next point is information and expression, meaning that a gene needs to contain a specific information. Like I was saying before, it needs to contain one specific information, and the transcription and translation of that gene is going to give rise to a specific expression. So this can be looked at as genotype, and this can be looked at as phenotype. So the information is what is contained at the genetic level, at the gene level, the information that is put into that specific gene, and that gene, when it is transcribed and translated, is going to give rise to a specific expression. Like if you take, for example, the same gene for height in case of garden pea, if the plant is going to have the dominant allele, then that dominant allele is going to get expressed and the plant is going to grow tall. So the information that is contained in that allele is for tallness, and once that allele is going to get expressed, the plant is going to grow tall. So that is information and expression. The last point is replication. Now genes are not fragments of DNA that are just flying around in the cell. They are part of the genome of the cell. So when the genome is replicating and being passed on to the next generation, that is the daughter cells, the gene needs to be replicated and passed on to the next generation. Unless and until it undergoes replication and gets passed on to the next generation, it cannot be called a gene. So these are the properties for a fragment of DNA to be called a gene. So a gene can be called a gene only if they have all of these properties. Since we are talking about molecular basis of inheritance, and that entails that we need to say that the chromosome, or rather the DNA, is the genetic material, that is the causative agent, that DNA is the one that contains the genes, and DNA is the one that is being passed on from generation to generation, and that it is the basis for what a cell is, right? So this is something that we already know, but back in the day that was not known. So people were still confused about whether nucleic acids, that is DNA or RNA, is the genetic material of a cell, or whether proteins are the genetic material of a cell, or whether lipids are the genetic material of a cell, or whether sugars are the genetic material of a cell. So they did not know that, and that led to different experiments that were conducted to figure out what is the genetic material of a cell. Now we are going to be talking about three major experiments here. The first one is the Griffith's experiment, which was done by Frederick Griffith in the year 1928. Now Frederick Griffith was trying to figure out a way to create a vaccine against Streptococcus pneumonia. At that time it's called as Diplococcus pneumonia, but currently we call it as Streptococcus pneumonia, which causes pneumonia, of course, and he was trying to figure out a vaccine for it. And with Streptococcus pneumoniae, there are two strains of the bacteria. There is a S strain and a R strain. Now S strain is called so because it is smooth, so it's the smooth strain, and it is smooth because it is encapsulated, meaning the cell is covered over by a layer of mucilage or slime, which is a polysaccharide. So mucilage or slime is just a polysaccharide which covers the exterior of the cell, and because of that reason the cell, the bacteria, when it is grown on agar medium, they appear glossy, shiny and smooth, and that's the reason why they are given the name smooth. The strain is given the name smooth. Also, one important thing to remember is that the smooth strain of Streptococcus pneumoniae is a virulent strain, meaning that it causes the disease. The other strain is the R strain, which is the rough strain, and it is rough because it is not encapsulated. So when this strain is grown on agar medium, it is not going to have that smooth glossy finish to it, and that is the reason why it is termed as a rough strain. Also, it is not virulent. So we have two strains here, one that is encapsulated and virulent and one that is not encapsulated and not virulent. So when Griffith was trying to do these experiments, he was using lab rats. So what he did first was to infect the lab rats with the S strain, the smooth strain, and saw that the mice died. So when smooth strain was infected, he got dead mice. Next, he infected the mice with the R strain only. When only the R strain was infected to the mice, he had living mice. The mice did not contract the disease, they did not die because of the disease, they were living. The next thing he did was to heat kill the S strain. So he took the S strain of Streptococcus pneumonia, heated it to a certain temperature, because of which the bacteria is going to not live anymore, and so this is basically dead bacteria. So he took the dead bacteria, which is heat killed bacteria, infected the mice with it, and he found that the mice were living. Okay, which is to be expected because heat killing a bacteria basically kills it, so it cannot be virulent anymore, so they are living. The next thing that he did was to take the heat killed S strain and then he added living R strain. So heat killed S strain, living R strain, and infected the mice again and found that the mice were dead. Now Griffith by himself could not understand or realize why the mice were dead if the S strain was already killed, meaning that it is not living anymore, so it cannot cause the disease, and R strain, even though it is living, by itself cannot cause the disease because it is not virulent. But he found that the mice were dead and he was perplexed. He could not understand why it is happening, and that was rather explained by other experiments that happened later on. But one thing that Griffith did notice was that when he took the dead mice here, which were infected with the heat killed S strain plus the living R strain, he found that the dead mice in their bloodstream had living S strain. Okay, so remember that the infection when it happened here had heat killed S strain, meaning that they were dead S strain, and living R strain. But when the dead mice were further looked into, their bloodstream was looked into, Griffith found that they were living S strain. So he could not figure out how that came about, but that was something that was explained later on. The next experiment that was carried out after Griffith's experiment was the Avery's experiment. Avery's experiment was carried out by three different scientists, Oswald Avery, Colin MacLeod and Macklin McCarthy, and in the year 1944. Now Avery's experiment is a continuation of Griffith's experiment because he used the same Streptococcus pneumonia strains. So he used the Streptococcus pneumonia strain S and R, and he used the same method like Griffith, that he heat killed the S strain, but after that it differs. So he took the heat killed S strain and homogenized it. Homogenization meaning that the cell wall was disrupted so that all of the contents of the cell would come out and become a homogenized solution. So bacterial cells were no more intact anymore. Now this homogenate was treated with different enzymes. The three different enzyme treatments that Avery did was with proteases 1, RNases 1 and DNases 1. So proteases are going to digest out the protein in the homogenate, RNases are going to digest out the RNA, and DNases are going to digest out the DNA. Once the homogenate had been treated with different enzymes, not that all of it was done at the same time. So in one experiment he would treat the homogenate with proteases, in another experiment he would treat the homogenate with RNases, and in another experiment he would treat the homogenate with DNases. Once the enzyme treatment was done, the enzyme treated homogenate which was missing one of the components, so it was either missing protein or RNA or DNA. This homogenate was mixed with living R cells. Now R cells are not virulent, so once the homogenate was mixed with the non-virulent R cells, this mixture was injected into mice. Like it was found in the Griffith's experiment, it was found that in one of the cases the mice was dead, in the other two cases the mice was not. So when the homogenate was treated with proteases, meaning that all of the protein in the cell or the disrupted cell, the homogenate, was digested out, so only the homogenate contains RNA and DNA, and this mixture is added with the living R cells and then injected into mice. Now when this was done, it was found that the mice were dead. Okay, so this explains that the transforming principle, meaning that how the R cells got converted to S cells, was not because of the protein in the heat killed S cells. Okay, the next experiment, he treated the homogenate with RNases. So the homogenate is going to contain just the protein and the DNA. Now this mixture again mixed up with living R cells, infected into mice, and found that the mice were dead again. So this proves that RNA is not the transforming principle. So not the protein, not the RNA, because both of them are not going to so even if RNA is not present in the homogenate, the R cell is going to get converted into the S cell, and it is causing the disease in the mice and it is killing the mice. So it is neither RNA nor protein. The third one that he conducted was with DNA, wherein he digested out the DNA from the homogenate of the heat killed S cells and mixed that homogenate with living R cells. So this homogenate is going to contain only proteases and RNA, and it does not contain DNA. So it's only going to contain protein and RNA, it does not contain DNA. And when this mixture was added with R cells, living R cells, and then infected into mice, he found that the mice were living. So this by itself proved that the DNA was the transforming material, meaning that the DNA has something on it which is getting into the living R cells, and because of that reason they are getting converted into S cells, meaning that they are synthesizing the mucilage around them, the polysaccharide covering around them, and they are becoming virulent. So there is something within the DNA that is doing this, which means that the DNA is the genetic material, or the DNA is the hereditary material. So Avery by himself has proved that the DNA is the hereditary material, and it is the one that carries the information from one generation to another generation. Another thing that Avery noticed was whenever this experiment was done and the R cell was converted into S cell, irrespective of how many times that R cell was made to divide and grow, how many ever generations they were made to grow for, the converted R cell remained as a converted R cell. So it did not change. It kept its converted S cell status as it is. It did not change over time at all. That we'll be talking about is the Hershey and Chase experiment. Hershey and Chase experiment was conducted by Alfred Hershey and Martha Chase in the year 1952. They used T2 bacteriophage and E. coli as their material for their experiment. Now T2 bacteriophage is like any other bacteriophage, a virus that is going to infect a bacteria. So bacteriophages are those viruses that infect bacteria. Basically they feed on them and they use the bacteria as a replicating machinery. So viruses by themselves cannot replicate, they need a host cell to replicate, and in case of bacteriophages they use bacteria as their host cell to replicate. Okay, so what they did was to label the T2 bacteriophage with either 35S sulfur, which is labeled with 35 meaning that it's the radioactive version of sulfur, or phosphate 32, which is the radioactive version of phosphate. So they labeled it with either radioactive sulfur or radioactive phosphate. Now, why exactly radioactive sulfur or radioactive phosphate? Bacteriophages are very simple creatures, in that unlike bacteria or other eukaryotic organisms, they do not have a very complex cell structure. Their cell structure is very simple. It is made up of a protein coat and DNA within it. So they're usually of a hexagonal head, and they have a tail, and they have sort of leg kind of things to attach to the bacterial cell, and they have DNA within themselves. So this will be the protein coat, and this is the DNA, which is the genetic material for these viruses. So when the virus needs to replicate, it is going to attach itself to a bacterial cell. It's going to create a hole in the wall of the bacterial cell, and the DNA is going to pass into the bacterial cell, and this DNA is going to replicate itself, and it's going to create the material for the protein sheath, and new viral components are going to be assembled within the bacterial cell. So the bacterial cell is going to give rise to many more viral particles. So all of these will be synthesized within the bacterial cell based off of just the DNA of the virus, which is going to enter the bacterial cell. So this is how bacteriophages work, and they're fairly simple. They're made up of just a protein coat and genetic material, which is the DNA. Now proteins have amino acids which have sulfur in it. So proteins are basically polypeptides. They are made up of multiple amino acids which are attached to one another, and proteins have amino acids which have sulfur as one of their components. And so if you're going to label with radioactive sulfur instead of the normal sulfur that is there in the protein coat of the bacteriophage, it's going to have the radioactive sulfur. So the protein coat of the bacteriophage is going to be radioactive. Why phosphate? DNA is made up of a backbone of sugar and phosphate. So if you're going to put in radioactive phosphate instead of the normal phosphate in the DNA of the bacteriophage, it's going to have radioactive phosphate. Now, as you most probably know, radioactive elements can be detected using autoradiography. And so if you have an organism that has something radioactive in it, we can simply extract out either the protein or the DNA, and through autoradiography we can figure out whether or not there is a radioactive element present in that component of the cell. Okay, so in Hershey and Chase experiment, they took T2 bacteriophage and then labeled it with either radioactive sulfur or radioactive phosphate. Once the labeling was done, they used these radio-labeled bacteriophages and then used them to infect bacteria. So either S was infected into bacteria or the P was infected into bacteria. Right? So it was either labeled with sulfur or phosphate, not both of them at the same time, and each of them were infected separately into different bacteria. Once the infection was done, right after infection, the bacteria were agitated to remove the attached bacteriophage from the bacteria. Right? So I showed you that the bacteriophage attaches itself to the bacteria, but when it is agitated, the bacteriophage is going to remove itself from the bacteria. So they were first agitated, both of them, and then they were centrifuged. So once you have agitated it, you're going to have bacteria separate from the virus. and when you are centrifuging them, because bacteria are heavier than viruses, they are going to settle lower than the viruses. So you can just take out the bacterial component only, and once the centrifugation is done, only the bacterial component was taken out and checked whether or not the bacteria within itself has either the 35S or the 32P. When they did the 35S labeling and did all of this, and after centrifugation checked whether or not the bacteria had 35S, it did not. So the bacteria did not have 35S, but 32P was definitely present within the bacteria. So it gave them a proof that it's just the DNA which is entering into the bacterial cell, and just the DNA which is carrying over the information for the bacteriophage synthesis into the bacteria. So Hershey and Chase experiment finally proved that DNA is not only the genetic material, but it also is the material that carries the information. So it's the hereditary material of any cell there. Now that we have figured out that DNA is the hereditary material and also the genetic material for any cell, we are going to study the structure of the DNA in detail. Now the structure of the DNA, or rather the components of the DNA, were studied by chemist P.A. Levine in the year 1920s, around 1920s. He's a chemist. He studied the chemical composition of DNA, and he figured out that DNA is made up of three basic components: sugar, phosphate, and nucleobases. Okay, so we are going to study each of them separately. Now the sugar that is found in DNA is the deoxyribose, or it's also known as the 2'-deoxyribose or pentose sugar, because it has 5 carbon atoms on the whole. The chemical formula is C5H10O4. It's a cyclic furanose structure, so it has a cyclic furanose structure. Now the numbering of the carbon atoms on this cyclic furanose structure start from the first carbon atom here, second, third, fourth, and the fifth carbon atom. It is known as the 2'-deoxyribose because on the second carbon atom it does not have a OH group but rather a H group. So all of the carbon atoms have one OH and one H group, one H, one OH group, like this. But in this case, this carbon atom does not have a OH group, which is the reason why it is called as the deoxyribose, and also from where DNA gets its name, which is deoxyribonucleic acid. Okay. In case of RNA, on the second carbon atom it will have one OH and H group, and hence it is known as the ribose sugar, and also the name derived from there, ribonucleic acid. Okay, so this is the structure of the sugar molecule, which is the pentose sugar that is found in DNA. Component of DNA is the phosphate group or the phosphoric acid. Phosphoric acid starts off in its unionized form, which is H3PO4. So you will have a phosphate group attached to 3 hydroxyl groups and 1 oxygen atom through a double bond. But once it gets ionized, it's going to lose all of its hydroxyl groups. It's going to have the formula PO43-. Now this is the molecule that is found in DNA and not the non-ionized form. So it's the ionized form that is found in the DNA, and this is the phosphate group. The sugar molecule and the phosphate group together form the backbone of the DNA, and the nucleobases form the ladder within the DNA. Moving on to the next component of the DNA, it is the nucleobases. Nucleobases are nitrogen containing components, and they form the bridge or the ladder, the steps of the DNA. Now nucleobases are basically of 2 types, purines and pyrimidines. Purines are double ringed structures, and they are of 2 types, adenine and guanine. Pyrimidines are single ringed structures, and they are of 3 types, thymine, cytosine, and uracil. Now adenine, guanine, thymine and cytosine are found in DNA. In case of RNA, it is adenine, guanine, cytosine and uracil. So instead of thymine, it is replaced with uracil. Now we are going to study each and every structure of each of the nucleobases. Starting off with the structure of the nucleosides, here I've written adenine, thymine and uracil. Like I was saying before, adenine is a purine, thymine and uracil are pyrimidines. The reason why I have written all three of them together is because adenine pairs with thymine in case of DNA and adenine pairs with uracil in case of RNA. Now coming to the structure, like I said, adenine is a double ring structure. It was discovered, or rather isolated, by Albert Koschel in 1885. It has the chemical structure C5H5N5. Its IUPAC name, that is its chemical name, is 9H-purine-6-amine. Its common name is 6-aminopurine. Moving on to thymine, it's a pyrimidine, so it's made up of one single ring. Its chemical formula is C5H6N2O2. Its chemical name is 5-methylpyrimidine 2,4,1H3H-dione. Its common name is 5-methyl-uracil. It was discovered, or rather isolated, by the same person, Albert Koschel and Albert Newman, in the year 1893, and they isolated it from the thymus gland of calf, and therefore they named it as thymine. Moving on, we have uracil, which is a demethylated form of thymine. So thymine has a methyl group attached here. Uracil has the exact same structure as thymine, just that the methyl group is missing in case of uracil. Now uracil has the chemical formula C4H4N2O2. Its chemical name is pyrimidine 2,4,1H3H-dione. It was isolated by Alberto Ascoli in 1900, but the name uracil was given by Robert Bannon. Now, why I have written these together is because it also gives you an idea as to how the bond formation occurs between adenine and thymine, or adenine and uracil. Now the bond formation between adenine and thymine is a double bond. So there are two bonds which are formed between adenine and thymine, or between adenine and uracil, and the bond formation occurs between the NH2 group here and the nitrogen group here, and the oxygen and the NH2 group here in case of DNA. In case of RNA, the bond is going to form between NH2 and N group here, and the O and NH group here. So there is a covalent bond that is going to be formed between the NH2O, N and NH here, or NH2O in case of uracil, and N and NH there. That is how the bond formation occurs between adenine and thymine, or adenine and uracil. Moving on to the other nucleotides that are there, guanine, which is a purine and hence has a double ring. Its chemical formula is C5H5N5O. Its IUPAC name is 2-amino-9H-purine-6,1H-one. Common names are 2-amino-6-hydroxypurine or 2-amino-hypoxanthine. They were isolated by Julius Bodo Unger in the year 1944. Coming to cytosine, cytosine is a pyrimidine and hence it's a single ring structure. Its chemical formula is C4H5N3O. IUPAC name is 4-amino-pyrimidine 2,1H-one, and the common name is 4-amino-1H-pyrimidine 2-one. It was isolated by both Albert Koschel and Albert Newman in the year 1894. Like I was talking with adenine and thymine, or adenine and uracil, wherein they have two covalent bonds between them, guanine and cytosine have three covalent bonds between them, and the groups which participate in the bond formation are the oxygen molecule here, the NH molecule here, and the NH2 molecule here. So all three of them together and combined with the NH molecule here, the nitrogen here, and the oxygen molecule here. So these 3 groups on guanine and these 3 groups on cytosine interact with each other and form 3 covalent bonds. So between adenine and thymine or adenine and cytosine you have 2 covalent bonds. Between guanine and cytosine you have 3 covalent bonds, and these are the groups between each of the molecules which interact with each other for the covalent bond formation.